Biology Chapter 19 Theory Video 5 Recombinant Human Insulin. Now human insulin is actually of very high demand because one form of diabetes mellitus is actually dependent on insulin. Because it is required to control the blood glucose level. Before genetically modified bacteria became available, people had to extract insulin from the pancreas of cattle or pigs. But in 1980s, they carried out different tests on this genetic engineering, and by 1983, recombinant human insulin became available. So the procedure is given in this diagram on 19.6. This diagram, if you study this diagram, you will understand how exactly it can be done. But to explain the process, first of all, what they do is it is not easy to identify and cut out the gene responsible for producing producing insulin in humans. So what they do is, instead of cutting it out, they take the mRNA for insulin from the pancreatic beta cells. So they express the insulin gene, and there would be large quantities of the mRNA for insulin. So then mRNA would be used as the template for reverse transcriptase to make a single-stranded DNA. Then that would be used as a template for DNA polymerase to make the double-stranded DNA, which is going to be the gene for producing human insulin. So this would be inserted into the plasmids that would transform the bacterium Escherichia coli or E. coli. Then the recombinant human insulin would be used in yeast or animal cells to produce this produce the human insulin that is required. So the advantage is actually recombinant insulin is going to be continuously supplied, whether there is a it would not depend on factors such as availability of pancreas in the meat trade, and also the produced insulin is not animal insulin, it is going to be human insulin, because there are differences between animal insulin and human insulin. So now there have been other advancements in the simple process as well. So now genetic engineers have given some changes to the nucleotide sequence of the insulin gene to give the molecules with a little bit of a different amino acid sequence. So they have different properties. Some will act really fast, some will act during a prolonged period of time. Because most of the common insulin actually should be taken right before a person eats. Then it would it would be in effect for about between 2 to 4 hours, and it will control the blood glucose level during that time. Then there is another recombinant insulin that would act for a period between 8 to 24 hours. So that will manage the insulin content, and thereby will manage the blood glucose level to produce to produce a balance or to maintain the balance of blood glucose level. So here, as you can see, the from the beta cells, mRNA is obtained, and using reverse transcriptase enzyme, it will be turned into a single strand of DNA, and then using DNA polymerase, it would be turned into the double-stranded DNA, right? So and after that, the process that I expressed would be placed in effect. Now, to write down a note on that before we go on to genetic markers. Recombinant human insulin. Moving on from obtaining DNA, obtaining insulin from animals like pigs and cattle, genetically modified bacteria, yeast, and animal cells are used today to produce insulin. mRNA for insulin from pancreatic beta cells would be extracted and would be used as the template for reverse transcriptase to make single-stranded DNA. Then the single-stranded DNA would be used as the template for DNA polymerase to make double-stranded DNA. This gene would be inserted into plasmids to transform the bacterium Escherichia coli or E. coli. Most modern insulin is made in modified yeast or animal cells because eukaryotic cells have Golgi bodies which helps the two polypeptides of insulin to be assembled and folded correctly. And also this results in other advantages such as making sure the supply is constant. Genetic engineers further have changed the nucleotide sequences of insulin to give molecules with slightly different amino acid sequences, producing insulin with slightly different properties. Some act fast and some act more slowly, and can be used by patients depending on their requirement. I would suggest that you draw the figure 19.6. That would make it easy for you to understand. Now to talk about genetic markers. Now when we continue to use pathogenic antibiotic resistant bacteria, antibiotics resistant genes as markers, using antibiotic resistance genes as markers is no longer practiced. So now what they instead what they do is they use enzymes that produce how we can identify the bacteria is by using enzymes that produce fluorescent substances. Now when you obtain enzymes from jellyfish, they make a protein called GFP, that is green fluorescent protein, So it would be bright green in ultraviolet light. So what we do is this gene for the enzyme would be inserted into the plasmid. So then when we shine an ultraviolet light, we can actually observe this. So we can use this same gene as part of the all the others as well. So that means if we when we add the gene that is needed for us to the plasmid, we attach this gene also, GFP gene also, we attach to the plasmid. So when the plasmid is taken up, we know that it would be presented or it would be expressed, and as a result we can identify them. Then another marker is enzyme beta glucuronidase. It is known as GUS. Again, this originates from E. coli. So if there is a cell transformed having this enzyme, if you incubate it with some colorless or non-fluorescent substrate, it will transform into colored or fluorescent products. So this will also help us to identify which ones have been taking up the genes. Write down genetic markers. Because of risk of creating pathogenic antibiotic resistant bacteria, use of antibiotic resistance genes as markers is no longer carried out. Instead, enzymes that produce fluorescent substances are used. So the gene for the enzyme is inserted into the plasmid along with the gene that we require. So when the plasmid is taken up, the bacteria will shine under ultraviolet light. Example, GFP protein, green fluorescent protein. Next one, under leave a line and write enzyme beta glucuronidase, within brackets GUS, from E. coli, when incubated with some specific colorless or non-fluorescent substrates, will transform into colored or fluorescent products. This is useful in identifying the activity of inserted genes in plants. Right, so here if you look at this in sundew, they are carnivorous plants, and they have on the left hand side is the transgenic sundew plant, which is expressing the gene for GUS. It has been placed in a solution of colorless substance, and the enzyme GUS has converted into dark blue color. So that means the plant has been genetically modified successfully. So that is what you need to understand. I hope you understood. If you have any questions, please ask. If not, please do question number 6.